All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So, this has been a mystery for me for the whole duration of the pandemic, that why are there individuals who may test positive and still are asymptomatic? And then we also know that there are individuals who were asymptomatic. We would then later on see that they may have antibodies, but they never tested positive because they never suspected they had the infection. So the um, basic point here is there are individuals who do not become positive, sorry, who do not become symptomatic. I remember a case where a woman sent me a message saying that my husband became positive and is ill and is on the bed lying down, cannot, exhausted, fatigued, and myself and my children are here in the same home and we have nothing. And she asked me what was the reason. I still remember it like a day that she asked me that, and I had no answer. So today we have an answer, and this is so exciting for me. And I wish that our healthcare agencies and research workers were looking into this first before looking into many other kind of uh, problems associated with SARS-CoV-2. So let's look at this one, this study. It is a study from UCSF. Here is the study. A common allele of HLA is associated with asymptomatic SARS-CoV-2 infection, July 19, 2023. So hot off the press. Although the individuals that are taking part in it, they were, I believe, up to March 2021. And the individuals who were the discovery cohort, and I'll explain that, they were unvaccinated. So this is unvaccinated individuals who became positive and they found that out accidentally. For example, they went to hospital for something else and they were testing positive as well, but they did not have symptoms. And I want to tell you the weakness of the or limitation of the study up front, that the study was conducted with a fawn-based survey, weekly survey of the symptoms. So with this, let's look at the study. Here are the references. This is drbeen.com. And I'm super excited today. We have moved to this library page, white theme plus all the lectures listed out here. Plus, we have just recently published and still are in the process of publishing 3D anatomy atlases with write-ups. Plus, now today, we moved to a white theme, back to white theme. We used to be white. However, I wanted to have white and reddish instead of blue. So we are here. Please forgive me if there are defects in this page, but we'll continue to improve this. And there is a link in the description if you wanted to get access to drbeen.com. This lecture is going to end up there as well, and then we'll apply for CME and offer CMEs on it. This is the reference. Then there are some other references as well, including the basic information about the terms that I'll use today. For example, what is an allele? What is a gene? What is a T cell receptor? What is major histocompatibility complex or MHC? What are HLA or human leukocyte antigens? I would explain them as well, but I have put those references for you too. So with this, let's start with my drawings. So these are gifts for humanity and they are continuing. So asymptomatic COVID, here is a possible reason. So you have seen that there are individuals who do not get symptoms. So imagine that someone, almost a majority of us, have become exposed to human coronaviruses before. About 86% of us have human coronaviruses in our throats. And Every year, the seasonal cold infections, 14 to 16% of those infections are caused by coronaviruses, meaning a majority of us have likely been infected by human coronaviruses and become ill with them in the past. And we still may have them in our throats. So, those individuals who have been exposed to these coronaviruses in the past, some of them are demonstrating that they're able to handle SARS-CoV-2 with such efficiency that they don't even develop the symptoms and they have already killed this virus off. And so the question, I hope that is in your mind now, what is in their system that does it? Why do they do this? And so I'll start giving you the answers. It's a long study. I'm going to give you the summary. T cell, cytotoxic T cell, in these individuals have a special major histocompatibility complex or HLA molecule on them. So all T cells have them. Actually, all nucleated cells have MHC1, and all antigen presenting cells have a MHC1 and MHC2. And I've discussed those in the past. These are little arms on the immune cells and nucleated cells with which these cells present whatever they encounter. So I'm going to actually stop sharing for a second and explain this. I have this one concept of MHC1 and 2 I've discussed for 3 years now. So I'm hoping that you are aware of it. So I'm just very quickly going to express this. Imagine I am a nucleated cell. Now, if I'm a nucleated cell, it is possible that I can get infected by viruses. Viruses like to get into the cells and replicate there or increase in number there, and they use the cell's machinery. When a virus enters a cell, so imagine I am a cell, the whole thing is a cell, and there is a virus in it. In the beginning, when the virus starts replicating, the cell's machinery is still not fully taken over by the virus because the daughters of the virus are not too many. So the cell still has a chance to behave and try to defend itself. So what it does is that cell will pick up the small pieces of the viruses that are being formed in it, and it will load them. Imagine an arm. It will load those pieces. Let's say this is a piece. Black piece on black glove will not work. Let's say my headphones. This is a piece of the cell, the virus, and the cell loads that piece on MHC molecule, major histocompatibility complex molecule. Then that cell pushes that molecule outside and shows to everyone that I've gotten something inside of me and this is what it is. This arm of the cell is called MHC1. When some cell are presenting things on MHC1, they are saying, I am sick. Inside of me is a virus. I am showing you a piece of it. Please help me. On the other hand, there are antigen presenting cells. There are professional antigen presenting cells. Professional antigen presenting cells, for example, macrophages, dendritic cells, B cells. What these cells do is that they look at their environment, they look at offending agents in the environment, for example, bacteria, viruses, other dead cells, dead pathogens, fungi. They pick them up, they eat them. That is the process of phagocytosis. Now remember this, these cells, professional antigen presenting cells, are picking up the pathogen deliberately because they want to clear it out. So it's a different function compared to getting sick with the virus and showing it the piece of it. So now the antigen presenting cell has eaten up the virus or pathogen or whatever bad thing is in the environment. It's going to shred it into smaller pieces. Then it is also going to load that small piece onto an arm or a molecule, and then it is also going to express that molecule on its surface to show it to other cells to say, hey, look what I got. you need to start fighting this thing. This arm of a professional antigen presenting cell that is presenting the antigen on it is called MHC2 molecule. So the summary: when a cell is sick, then it presents the antigen on MHC1. When a cell is not sick, but is just attacking the pathogens present in the surrounding and killing them, shredding them, and then showing the pieces out, that would be presented on MHC2. Now, if there is a cell that is an immune cell, nucleated cell, able to present on MHC2 but got sick, for example, HIV causes T cell or the immune cells to become sick, right? Helper cells. Similarly, there can be other viruses, including SARS-CoV-2, that can infect the professional antigen presenting cells. In that case, professional antigen presenting cell, for example, macrophage or dendritic cell or B cell, will present the antigen on both arms. It will present it on MHC2 as its function to present something that is present in the environment, and it would also be presenting the same thing on MHC1 to say I am sick as well. So this is one important thing to keep in mind. And we have then done this discussion that when the antigen is presented on MHC1 or 2, then the B cells and the T cells and the other cells, they will come and they will check it out. And they'll say, oh man, you seem sick. Or, oh man, you're presenting us something that is not really nice. We are going to go and attack it. So this is how the innate arm cells activate the acquired arm cells. So this is a quick refresher of the past discussions. Now going back to our presentation. And I am so excited about today's talk, and thanks to these researchers for doing this research. Okay, so we now know that some individuals, almost all of us, have, I'm saying almost all of us, have previous exposure to coronaviruses, human coronaviruses, and then some of us are able to handle SARS-CoV-2 without even getting symptoms. Okay. Cell. So here, I said MSC cell because I saw my camera wasn't working. I hope it is working and audio is working. So not MHC cell, the T cell. Imagine that this is a cytotoxic T cell. We also have done this discussion in the past. We have helper T cells and we have cytotoxic T cells. We have regulatory cells and we have alpha, delta, or other cells too. Here is this T cell. This T cell is presenting, look here please, it is presenting a piece of the pathogen. Imagine this is SARS-CoV-2 or any other coronavirus, and this is a piece of the spike protein, one tiny piece of the spike protein loaded on this MHC1 and presented on the outside. If it is a cytotoxic T cell, then it has perforins as well, and it has granzyme as well, and it would use them to attack the other cells, the infected cells, and kill them. But here, that is not really the important thing. What is important is it is a T cell, and this T cell is presenting the piece of a coronavirus spike protein on its surface. Now, here is the important thing to note. After this, the rest of the study is just simple. This is the key part. They found out that people who have a gene called HLA B1501. What does that mean? Human leukocyte antigen. And what does that mean? I'm going to skip ahead for a second and show you what does this gene mean. So here, on our chromosome number 6, of course, chromosomes have two arms, right? So petite arm or small arm and a big arm or Q arm. The petite arm, chromosome number 6, location number 21.31, has a set of genes present there, which are called human leukocyte antigen genes or HLA genes. These genes encode MHC1, MHC2, and MHC3, or HLA class 1, HLA class 2, and HLA class 3. These are the classes we are talking about. MHC1, as I mentioned before, becomes an arm to present an antigen to the surrounding inflammatory cells or immune cells. MHC2 is also an arm, or HLA class C or class 3 is also an arm with which the professional antigen presenting cells present the antigen to the environment. Now, another quick thing here, if you're going to read the study, you will see that the class 1 or MHC1 is further divided into type A, B, and C. And we are talking about B here. So this is the exact gene that we are discussing. We are discussing MHC1 gene, and then type, or within that MSC1, the type B gene, and then within B, a subcategory of 1501. There will be a mention of class 2 as well, but only as a supporting role. Class 1 is the primary discussion today. So now if I go back to the discussion, sorry, I should have put that over here. So now here, in some individuals, there is a HLA B1501 allele. That means in that area of the chromosome, the amino acid sequences that give rise to the protein that it makes, MHC1 arm and MHC2 and 3, those sequences can be different, slightly different, because of which the molecule, the arm that they make is different. It is just like you have a different arm and I have a different arm. We still have the arms. We still have things that are called arms, but they may have slightly different structure. Now imagine that the binding area, my hand, is going to be entirely different from your binding area, your hand. That doesn't happen in humans. We all have similar hands. But the T cell receptors and immune antibodies by B cells, they have different binding regions. Now, this arm in some individuals is unique. Why is it unique? Pay attention to this one, please. It is unique that when this virus is present in these individuals' T cells, then this unique gene allele makes an MHC1 molecule which specially gets attracted to a special part of the spike protein. So you would say, fine, it attaches to a special part of spike protein, so what? Here is the important thing. That special part is very similar, or as we would say in technical term, conserved, across human viruses and SARS-CoV-2. So here, look at this little red block. Imagine this is a piece of the spike protein that this MHC1 is binding with. In the SARS-CoV-2, this piece to which a T cell and MHC1 in these individuals bind is called NQ-KLIANQF, or in this study, they simply called it NQ8. So here, NQQF. And in the human coronaviruses, the same piece on the spike protein with just one amino acid difference is called NQ-KLINAF. There's just one amino acid difference. Instead of Q, there is A. So in these individuals with this genetic makeup, when they react, these individuals and their immune system, their T cells, when they react to human coronavirus somewhere in the past, before the pandemic, they develop cells that attack a conserved part of the spike protein, a special part of the spike protein. Interestingly, that part is conserved in SARS-CoV-2 as well. So now when these T cells are their memory T cell, and then comes SARS-CoV-2 and says, I'm going to infect you and I'm going to do bad things to you, their memory T cells jump into action and they attack the same conserved part and very quickly take care of these SARS-CoV-2 viruses and kill them. You don't even find out that this happened. That is the study. I love this study because of this is the discovery that was needed to understand why people are asymptomatic. So back here, there is a part on this spike protein called NQ, and AF in human coronaviruses. People with this genetic makeup of MHC molecule, their MHC molecule specially stabilizes and attaches to that part of the spike protein and presents it. Then those cells become memory cell. In the future, when SARS-CoV-2 arrives, for majority of us, SARS-CoV-2 is a new foreign pathogen. It's deadly for us. But for these individuals, it's nothing they've not seen before. So that one part on the spike protein of SARS-CoV-2 is very similar to human coronaviruses, and these memory cells will wake up and bind there and just kill it. I just repeated myself. Okay. So the details. Researchers say, Kimi Nature says, did it say Q and A are amino acids? So these are kind of representation of the amino acids. So details. 20% of the population remain asymptomatic according to this study. I believe a larger number, but anyways, they said, here is a study which shows 20% of the population remains asymptomatic. The postulation, this research group, they postulated that maybe there is something to do with the HLA variation that helps with this. Again, that doesn't mean that they somehow just had a very strong dream about it. Researchers would postulate and they'll think in so many ways. So one of the ways they thought was maybe it has something to do with the HLAs, because in the past there have been studies that had been saying that, hey, this may be something to do with the HLA, and I'll show you the example of that. So here, if you see, this is another study. This was 2020 June. CD4 positive T cells support polyfunctionality of cytotoxic CD8 cell with memory potential in immunological control of tumor. And here they also have HLA, the same gene, which is different. So the folks who are research workers, they have seen such things happen before. So they say maybe this is involved too. So then what they did was they enrolled 29,947 individuals who may have given bone marrow in the past. And so their genetic makeup of their cells was known. So they had a very high resolution HLA genotyping of these individuals. So researchers reached out to them and said, hey, we already have your bone marrow samples. We already know your genetic genotyping of your cells. Will you be willing to help us to also now tell us what are your symptoms, and if you become positive, when do you become positive and what are your symptoms? And so these individuals said yes. And then they started tracking them on a weekly basis. Turned out there was a discovery cohort. There were a number of patients who said, hey, we became positive. These were 1,428. Now, when they became positive, then the next question to them was, did you have symptoms? And some of them said, I became positive because I went to the hospital for something else and I got positive test as well, but I don't have symptoms. And they had a number of other criteria as well to figure out if they were positive without symptoms. And they laid down in their method and design. I'm just giving you one example. So these 1,428 individuals were unvaccinated. They reported being COVID positive. And then out of them, some of them said, we didn't have symptoms. And when they looked at their genetics, they found out that they had a strong association with HLA B1501. So that is how they found that there is an HLA involvement. Of course, then the next step was to figure out, is it true or is it just a fluke? So what they did was they repeated it with, they had another cohort from Europe and they had their samples as well. Then they repeated it in those samples. They repeated it in other cohorts. And so they kept verifying. Then what they did was they built this peptide, this piece of spike protein, and then they handed that spike protein piece over to the T cells from these individuals, some of them, not all of them, and they saw the reaction as well. So they did multiple ways of verifying. And so they have a statistically significant association. Still, it's not the only reason that somebody could be asymptomatic. There may be other reasons, but this is one that has now been proven with statistical significance, this association. So, what they found was, after all of this research, and I'm going to repeat now, that SARS-CoV-2 in some individuals who are HLA B1501 becomes easy to kill because its spike protein has a conserved peptide on it called NQKLIANQF that is easy to be identified or attached with in these individuals because they have learned to attach with this in the past coronaviruses. So then your question will be, what about others? If I get coronavirus and I'm not HLA B15, what happens to me? Well, your viruses, your cells will attach to this human coronavirus as well, but maybe somewhere else. Because of that, your and my T cells will not develop a memory set of cells which can attack this conserved part. And so it is possible that these memory cells that are present in you and me who are not, let's say, HLA B15, those memory cells do not recognize anything on the SARS-CoV-2. They're not cross-reacting. And so even when the SARS-CoV-2 comes in, these T cells keep sleeping. They say, well, we have nothing to do with this thing. And that is why for us, it's altogether a new virus. And that is why it is able to cause damage. The other thing they found was that these T cells present in HLA B1501, they were cross-reacting, meaning they had previous human coronavirus and now they are able to connect with SARS-CoV-2. Secondly, they were highly polyfunctional. They were able to also release cytokines. Many times the cell is either doing more cytokines and less presentation and so on, but these are polyfunctional. They were able to do cytokine production as well and various kinds of cytokine productions. And then they were memory as well, and they were able to attack SARS-CoV-2. Beautiful cells. And this I have already discussed, that this was the genetic structure. Now, what was the result? They saw that those who were asymptomatic, the frequency of this gene, HLA B1501, was 0.1103 versus those who didn't have this gene, they had the frequency 0.0495. Or those who became asymptomatic, they had a lower frequency of this gene. The odds ratio, 2.38. So 2.38 times less possibility, or 2.38 times more odds of being asymptomatic. 95% confidence interval, 1.51 to 3.65. So range is odds of remaining asymptomatic of those people who are HLA B1501 was 1.51 times to 3.65 times. And p-value was quite significant, or 3 multiplied by 10 to the power minus 5. Then there is one more interesting thing that I think is, you're going to like this one. Those individuals who have this gene, HLA B1501, from mother and from father, meaning they had two copies of this gene, these individuals were more than 8 times less likely to be symptomatic, or more than 8 times more likely to be asymptomatic. That's the right way to say it. Now, in the same study, there are some other interesting things. So, if somebody's thinking that, hey, that may be then another genetic makeup for someone who becomes more vulnerable to COVID. And they found that too. They said individuals with HLA B4601 have, and these are other studies that show it, and they were referring to it, that have a lower predictability of binding with SARS-CoV-2. So the T cells in these individuals, old T cells, memory T cells from other coronaviruses, have lower predictability to bind with SARS-CoV-2. And because of that, these individuals are more vulnerable to SARS-CoV-2 because their other coronavirus memory cells just do not recognize this SARS-CoV-2 at all. Then, final point that is also interesting, they also found that individuals whose gene for MHC2, remember MHC1 and 2? MHC2, the gene Drbeen if there were individuals with this MHC2 gene, then they saw that this gene actually enhanced the effect of 1501 MHC1 gene. So MHC2 played a supporting role for MHC1 if the genetic makeup was the following. That is also very interesting. So this is the discussion. What is interesting for me is that I used to say in the beginning of the pandemic. So let me back up for a second. There is a lot more in this study. There is a lot to be studied and read in this study. You would love it. I just gave you a summary and an idea of what this study is. The method, the design, the data, everything is good. The questionnaires, it's very interesting. Now, I used to say in the beginning of the pandemic that our time, our being the healthcare research workers' time, agencies, their time, resources should be spent on figuring out people who are asymptomatic, why are they asymptomatic? Because we could then learn lessons from there and apply them for others. For example, here we found out in this study that HLA B1501 attack a specific part of the spike protein which is conserved across coronaviruses, that NQ8. Why not, if we had to make a vaccine, then why not make a vaccine that attacks that part? Why not make therapies that attack that part? Right? So instead of spending 3 years just looking at other things, if the focus was on asymptomatic individuals and see why they're asymptomatic, I think we would have had a better chance of managing this pandemic in a better way. So, anyways, then I have one more thought in my mind, that is there any relationship of this genetic makeup with vitamin D levels? So maybe these individuals also have better vitamin D levels too. So don't know. Then there is another question in my mind, that these individuals, how far back did they become infected? Because they said pre-pandemic, but again, this study's time is up to March 2021. So pandemic to March 2021 is almost a year. So it's not 3 years. So the question is, how long these memory cells continue to help? So at least in this study, we can say that if somebody was infected a year before from normal human coronavirus and they had this genetic makeup, then even a year after, those memory cells would cross-react with SARS-CoV-2 and protect them. That's a very interesting thing to note. Can that continue? How long will that continue? That is also a very interesting thing to note. And so these are a few things that are interesting with this study. But for the first time, there is a demonstration of people who are asymptomatic, we knew that, and why they may be asymptomatic. At least one reason is in front of us. So thank you very much. Please like, subscribe, and share. There are links in the description if you like this work. This work gives me goosebumps when I look at these researchers and their work, and when I prepare it for you to present it. I love to simplify and draw these things and present them. If you would like to support this work, there are links in the description: Substack, Patreon, YouTube, Dr. Bean, and join me for that. So, with this, have a great weekend. Share this one. If you know someone who became COVID positive and didn't have the symptoms, share it with them. If you are one, I think there was a cool bean, her name was Mary. She said that she had this. She's here in the, hopefully in the comments now. So that is a discussion. Meeple Art says, can you post a Discord invite for those that would like to discuss? So, meaning Discord server. Can I request John? John may be here as well. John, can you please present Discord server link? Maybe if not now, then next. Mary, you are here. I tested positive 323, but was negative any previous times. Very interesting. So the question is symptoms or not. Testing positive is all of these did, but they didn't have symptoms. So interesting as well that they were unvaccinated. They didn't have symptoms and they were positive. So very interesting study. I would request you to give it a read. And yes, Mary says I'm here. Mary, this study today was for you. So with this, thank you very much. Have a nice weekend. I would see you on Monday, actually, maybe next week, not on Monday, but some other days. And thank you very much for the drawings. I spent a lot of time today drawing these because I was just so excited about this study. This is, for me, there've been a lot of vaccine veinings and memory cells and T cells and the spike going here and spike going there and the toxicities. This is the first good study in the asymptomatic world that can be used for others as well. So I really love it. Thank you and have a good night.